Hello, hi. We have started our survey on fungi module in the previous sessions. Today we'll discuss about MUCOR fungi. Before we move into the details of the topic, let us see a few slides. So this will be the order in which we'll be proceeding through the fungi lecture series. We have to see the life cycles under Zygomycota, that is Rhizopus and MUCOR. Then we'll be seeing around 5-6 life cycles under Ascomycota. After that we'll be seeing Basidiomycota. Zygomycota, Ascomycota, Basidiomycota are examples of true fungi, and after that you have to see a fungi-like organism, that is the phylum of Oomycota. We have to see 2 life cycles under this. Then we will be seeing about lichens, mycorrhiza, fungal toxins, etc. So let us move into today's discussion, that is discussion about MUCOR. So last class we have seen everything about Rhizopus, right? Rhizopus, but here you can see that it is almost similar. Both were having, even Rhizopus was having this prostrate system, then an erect hypha, then at the tip of the erect branch presence of sporangium which carry sporangiospores, etc. Here also you are seeing both. Rhizopus is also called a bread mold. Even this particular MUCOR is also called a bread mold. So there are many similarities because they are coming under the same order of MUCOR release, but there is a few small fine differences that we will see today. And you can see that this particular MUCOR was famous during the corona days because almost during the same time there were spread of diseases called mucormycosis because of the fungal infection caused in human and animals, etc. You can see that it is also called pin mold because at the tip of the sporangiophore you will be seeing black colored sporangia which are like pinheads. So that's why it is also called pin molds as well. Then you can see that rhizoids are absent in this particular prostrate mycelia, right? But in the case of Rhizopus we have seen this, so we will see such differences as well. So let us move. These are the differences between MUCOR and Rhizopus. You just see visibly also many things you can observe. From one particular node, in the case of MUCOR only one sporangiophore arise, but in the case of Rhizopus from one particular node there are many. It's like a tuft, T-U-F-T-S, tufts. Okay, from one particular node many sporangiophore arise at the tip of which sporangia is carried. So this is one difference. Then in the case of this particular Rhizopus there are 3 differentiation of thalli. One is an erect sporangiophore which carries sporangia. Then there is a prostrate stolon branch. Okay, then at that below the stolon branch you can see the formation of rhizoids. This will be carrying absorbing all the nutrients, etc. Then in the case of MUCOR you can see that rhizoids is absent, just prostrate and erect system. There is no presence of underground rhizoids. This prostrate branch will be absorbing the nutrients from the medium or substratum. Then this is the like second or third difference. The fourth difference is that in both in MUCOR and Rhizopus we will see the formation of zygospore during the sexual reproduction, but in the case of like Rhizopus, after this zygospore formation there is a resting period. When the zygospore is formed from this septum, and from this septum it will be getting detached from the mycelium and it will be laying as such in the soil or the substratum for a long time, and during the time of reproduction when it is almost ready to reproduce, at that time only meiosis happens within this particular zygospore and then a promycelium will be formed from this. But in the case of MUCOR the resting period is after meiosis. Okay, after the zygospore is formed, then initially meiosis will happen. After that only the resting period happens, and after the resting period promycelium develops. So this is one difference. So these are the main differences. And one more last difference if you want to write, in the case of dehiscence, after asexual reproduction you will see that there is a sporangial formation at the tip in the case of Rhizopus and in the case of MUCOR. But during the dehiscence of the sporangial wall, in the case of Rhizopus, initially all spores will be carried away by the wind, but in the case of MUCOR it is like something like sticky. It will be sticking at the tip of this particular columella. Okay, there is one structure called columella. We will see that it will be sticking to this columella, and slowly only like wind will be carrying these spores. So these are the main differences between rhizopus and MUCOR. Apart from this, almost everything is similar. Let us see. So it belongs to kingdom Mycota, like fungal kingdom. Then division Zygomycota, subdivision Zygomycotina. Okay, class Zygomycetes, order Mucorales, family Mucoraceae, genus MUCOR. These much things you have to write. The subdivision you can ignore. Apart from that, you have to write every answer related to any genus. Then in IFS, this particular information may be of importance. So in the case of anaerobic liquid culture, if you are growing this particular MUCOR in anaerobic conditions, where carbon dioxide is in excess other than oxygen, okay, oxygen is absent and carbon dioxide is present, in that the hyphae break and forms yeast-like bodies called torula. Okay, it's called torula condition. Yeast-like bodies called torula. When the oxygen is available, it will be reverting back into the filamentous form. Okay, just like mycelia and hypha form. Okay, so just imagine that during unfavorable anaerobic condition, the fragment will be breaking to form yeast-like bodies called torula. Okay, torula condition it is called. So after the oxygen is available, it will be again coming back to mycelia and hypha form. So in the case of when you are drawing this particular MUCOR thallus, you have to keep in mind that from one particular node only one sporangia 4 is arising and it is erect. Okay, so this kind of habitat these are same for both rhizopus and MUCOR. Okay, organic soil or dead and decayed organic matter or jam, jelly, bread, etc. Around 50 species are known worldwide. Even it is infecting animals and human beings through a disease called mucormycosis. It will be affecting lungs, eyes, etc. It's a very fast growing fungi, and in the case of young fungi, young MUCOR, so the body will be generally cenocytic and aseptate. There are no septa in young mycelia, but in some species when it is mature, you can see septa formation, and it is generally cenocytic. Cenocytic means what? Without septa, many nuclei. Okay, the nuclei is there. Okay, when the zygote divides, okay, many nuclei are formed, but no septa is formed. Okay, it is not divided into multicellular fashion. It is just a tube-like body with many nuclei. That is called cenocytic. So, and the cell wall is made up of chitin and chitosan. This cell wall is made up of chitin and chitosan, not cellulose, right? So characteristics of MUCOR, all things I have explained. They are saprophytic. What is coprophilous? Coprophilous means when it is growing in the cow dung or any other dungs, then it is called coprophilous. So other things we have explained, and there is one more thing. The vegetative body of MUCOR is eucarpic. Eucarpic means what? If the thala is differentiated into vegetative structure, prostrate structure, and erect structure, fertile structure. So when the thallus is differentiated into vegetative and fertile structure, then it is called eucarpic. So that is one thing. Yeah, these are the characteristics. And major food material is in the form of glycogen and oil droplets. Okay, glycogen and oil droplets. It's not starch. Okay, then this is the structure of MUCOR. I have almost explained everything. One thing, as we are moving to the asexual reproduction, I will explain a few things here. During the time of asexual reproduction, you can see that one particular like hypha will be specialized to form a sporangiophore to carry sporangia. And when the sporangia is getting formed, the entire cytoplasm from the hypha, from the sporangiophore will be moving to the tip of the hypha, and it will be getting swollen in shape, okay, because of the accumulation of cytoplasm and many nuclei. So this is the first step. You can see that the cytoplasm and nuclei flows towards the peripheral region. The tips of the sporangiophore swells up. You can see the tip getting swollen to form a spherical sac-like structure called sporangia. So when they are asking a question about structure of Zygomycota or structure of MUCOR, this is the way you have to write. The tips of the sporangiophore swells up to form a spherical sac-like structure called sporangia. Now the protoplasm and nuclei gets collected or accumulated at the peripheral region. As it is getting swollen, this cytoplasm and nuclei gets collected or accumulated at the peripheral region. Okay, so what happens to the other central region? Small vacuoles, okay, small vacuoles, air bubbles type small vacuoles forms a dome shape at the base of this sporangial wall. Okay, So the top region becomes sporangioplasm, okay, where the spore forming material is accumulated, and the bottom area becomes columella-plasm, which will be forming columella. So after the accumulation of small vacuoles in the bottom of the sporangial wall, there will be a septa formation by separating sporangia and columella. Okay, so both are formed. Now this sporangioplasm undergoes progressive cleavage. Okay, this kind of terminology you have to use. Progressive cleavage, that means the cytoplasm, just like I said during the rhizopus explanation, imagine it's like a cake. Now you are like cutting into small pieces, and each of the small piece of cytoplasm will be having many nuclei. Then it will be developing a wall around itself, and it will be forming sporangiospore. So this is again the structure of mucor. If they are asking a question on the structure of any genus, this is the way you have to write. You can write about the mycelium. These are highly branched, fine network of hyphae. These are filamentous in nature, aseptate or sinusotic. You can write about the sporangiophore, elongated, erect and fertile. Then you can write about the columella, and it is dome shaped, etc. You can write. Then you can write about the sporangium. Okay, sporangium means what? Like from the sporangiophore, the tip will be swollen and it will be forming sporangium. These things we have already discussed. Then you can write about the spores as well. Now coming to the life cycle of mucor, you have to write about the vegetative reproduction, asexual reproduction and sexual reproduction. Vegetative reproduction is through fragmentation. Asexual reproduction is through aplanospores and chlamydospores. Aplanospores is also called sporangiospores. We have seen already in the previous slide. Then chlamydospores. Then sexual reproduction is through gametangial copulation, which is almost same like the rhizopus. So vegetative reproduction, simple. The body gets fragmented and each fragment develops into new vegetative cell. Asexual reproduction is through these three methods. Okay. Sporangiospores means what? I have already explained how the sporangiophore develops into this particular sporangium, and within the sporangium you can see the spore forming region and the columella region, and this particular cytoplasm, protoplasm gets divided into multinucleated protoplasm. It will be developing small walls around itself, so it will be forming sporangiospores, and during maturity sporangiospores will be released outside by the dehiscence of the wall of the sporangium. So during favorable condition, germ tube will be formed and it will be forming the vegetative hypha. Now this is the thing. Okay, the hyphal tip became swollen, then separate septa formation between columella and the sporangia, and the cytoplasm is divided and multinucleated. This particular each nuclei will be carrying some cytoplasm around itself, and it will be forming sporangiospores. Okay, then it will be after the sporangial wall is open, this particular spores get stick to the columella and it will be released slowly. It will be released. So I have discussed everything here. You can pause the video and take the notes. Then chlamydospores. Similarly, in the case of rhizopus, one particular intercalary region of the mycelium gets septated and it will be aggregating cytoplasm and nuclei, and it will be detached from the body. During favorable condition, new germ tube will be formed, which will be forming into a new mycelia. In the case of oidospores, when the mycelium is growing in the substrate which is rich in sugar, for example a bread, so a pearl-like reproductive structures will be formed at the tip of the mycelium and it will be detached. So then it will be germinating. This particular reproductive spore-like or pearl-like structures will be germinating into a new mycelia. Symbol. Sexual reproduction is same as the case of rhizopus. You can see that there is an initiation of progametangia. Then the progametangia become gametangia. Then this gametangia will be, after the fusion of nuclei, will be forming a zygospore. This is the two walls of zygospore, exospore and endospore. From the zygospore, a mycelium will be, promycelium will be released at the tip. You can see the formation of zygosporangium. This zygosporangium will be forming carrying spores. Okay, these spores will be two types of spores, plus spores and minus spores, because this is formed by the fusion of two types of thallus. Okay, plus and minus strains. So this is all about the notes. You can pause the video and take the notes. Okay, I have attached every details in the successive slides. So this is an extra note, same sexual reproduction note, but from a different source. I found that this is giving more explanation. You can pause the video and take the notes as well. So it is very simple. So two gametangia, like two thalli came together, formed a progametangia, then gametangia, then similar like remaining things we have already explained. Thank you so much.